Okay, respiration, part 3. This is the Krebs cycle, and we are here. It's also called the citric acid cycle, you'll notice. Why that is, I'll discuss in a little while. Now, before we move on with the Krebs cycle, what we've been doing up until now, and including the Krebs cycle, so the whole point of glycolysis and the link reaction and the Krebs cycle is to, release the energy from glucose. Okay, and the way that we are doing that is we are collecting electrons. So every time the molecule of glucose or the products of the breakdown of glucose are dehydrogenated, every time they're oxidized, we produce a molecule of reduced NAD. And that reduced NAD carries electrons, and those electrons have all got energy. Okay? So every time we produce a molecule of reduced NAD, we've got these electrons attached. And the whole purpose of these first 3 stages really is to collect those electrons. And what needs to happen is they need to go to the electron transport chain, and that's where ATP is produced, or that's where the vast majority of ATP is produced. The glycolysis and the Krebs cycle actually produce enough energy, or release, sorry, we always use the word release enough energy to produce a couple of molecules of ATP, but really sort of insignificant quantities. The whole purpose is to collect reduced NAD, and that reduced NAD is going to carry the electrons, because remember, NAD is an electron carrier. It's going to carry the electrons to the electron transport chain, where they will be used to phosphorylate ADP. Okay? So that's just sort of like a little explanation of where we are up until this point. Now, the Krebs cycle is where the vast majority of the NAD reduced is produced. Okay? So what goes on in the Krebs cycle? Let's have a look. And that's what our learning outcomes are for today, to describe the 3rd stage of respiration, which is the Krebs cycle. So it happens in the matrix of the mitochondria, like the link reaction. It's the 3rd step of respiration, and it requires oxygen, like the link reaction. Remember, the only stage of aerobic respiration that doesn't require oxygen is the glycolysis that happens outside the mitochondria. So what does the Krebs cycle do? Well, it does loads. Okay. And we're going to have a look at this. And I really, truly recommend that you spend a little bit of time going over this, looking at different resources, finding out what's going on here, or just consolidating what's going on here. So at the end of the link reaction, you had a 2-carbon compound which was attached to a coenzyme. Okay. That 2-carbon compound is carried by the coenzyme to the Krebs cycle. The 2-carbon compound then joins the Krebs cycle, and when it joins the Krebs cycle, it actually joins to a 4-carbon compound, which produces obviously a 6-carbon compound. Really simple math there. 2-carbon compound, add a 4-carbon compound, get a 6-carbon compound. So now we're in the Krebs cycle. The 6-carbon compound is decarboxylated, a carbon is removed, and it's oxidized or dehydrogenated to produce reduced NAD, 1 molecule. So now we've got a 5-carbon compound. This 5-carbon compound is decarboxylated, removing a carbon, and it's oxidized or dehydrogenated to produce another molecule of reduced NAD. At this stage, there's enough energy released to produce 1 molecule of ATP. So now we've got a 4-carbon compound. This 4-carbon compound has to be further modified before it's ready to cycle to the beginning again. And what happens is it's oxidized or dehydrogenated twice, and that produces 1 molecule of reduced NAD and another molecule of reduced FAD. Okay, FAD is just another enzyme carrier, and we don't need to worry about it apart from to know that it carries electrons until we get to the electron transport chain, and then you will see why FAD is different to NAD. Okay? So now we've produced all of the reduced NAD and FAD we need. We've produced a little bit of ATP, and we have broken down all of the bonds in our glucose molecule and its products. There's nothing left. There's no energy left in that original glucose molecule. And our 4-carbon compound is ready to receive that 2-carbon compound from the link reaction again. Okay? I'm sure this looks really very complicated at the minute. It looks simpler when you look at it in a cycle form. Okay, because it is a cycle, it makes much more sense. It's a cycle because the 4-carbon compound that joins with our 2-carbon compound is recycled at the beginning so it can receive another 2-carbon compound. Okay, so let's have a look at this. This is what you need to know about the Krebs cycle for this course. Okay, so our carbon count, 2 becomes 6 and then becomes 4. So what do I mean by that? So our 2-carbon acetyl compound that was produced in the link reaction is attached to a coenzyme, coenzyme A. And the whole purpose of coenzyme A is to carry the 2-carbon acetyl compound to the Krebs cycle. Once the acetyl compound binds with the 4-carbon compound, as it does here, to produce citric acid, the coenzyme is released. It's never used up in these reactions, like any enzymes, and it will then cycle back to the link reaction to pick up another acetyl compound, which it will then bring to the Krebs cycle, drop it off, and cycle back again. So you've effectively got a little cycle going on up here as well. So our 2-carbon acetyl compound is brought to the Krebs cycle, where it is released so it can bind with a 4-carbon compound. The 2-carbon compound and the 4-carbon compound combined produce a 6-carbon compound called citric acid. So this is why the Krebs cycle is also called the citric acid cycle. It's called the Krebs cycle because Mr. Krebs or Dr. Krebs or Professor Krebs was the first bloke that actually discovered it, but it's called the citric acid cycle because the citric acid is the first product of the cycle. That's where it all begins. Okay, so this 6-carbon compound is decarboxylated, the carbon is removed, and that produces carbon dioxide. And remember, if you think about that really, really simple equation that we used to talk about when we talk about respiration, carbon dioxide is one of the waste products of respiration. This is where it comes from in the Krebs cycle. It's also oxidized or dehydrogenated, so we produce 1 molecule of reduced NAD. And this is in pink here because the reduced NAD is heading off to the electron transport chain. So now we've got a 5-carbon compound, and that 5-carbon compound is decarboxylated again. So another carbon is removed, giving us a 4-carbon compound, and that carbon also produces carbon dioxide. Okay, so carbon dioxide, remember, is a product or a waste product of respiration. Okay, in this reaction, the 5-carbon compound is also oxidized or dehydrogenated, producing another molecule of reduced NAD that's going to go to the electron transport chain. And at this point, there's also enough energy released to produce 1 molecule of ATP, okay, which is going to just be used by the cell. So now we've got a 4-carbon compound, but we're not finished with it yet. It needs to be oxidized or dehydrogenated twice more before it's ready to receive the acetyl group again. And those 2 oxidations or 2 dehydrogenations produce 1 molecule of reduced NAD and 1 molecule of reduced FAD, both of which will go to the electron transport chain. So at this point here, we have released all of the energy from glucose. Okay, there's nothing left. Most of it, we've produced a little bit of ATP, but most of it is tied up in all those electrons that are being carried mostly by NAD and occasionally by FAD. And those electron carriers, FAD and NAD, are going to go to the electron transport chain, and that's where the magic happens. That's where we produce all of the ATP that we need. Okay, very important down here. Remember, all this happens twice for every glucose molecule entering glycolysis, because the link reaction produces 2 molecules of acetyl-CoA because the glycolysis produces 2 molecules of pyruvate. Okay, so that's important. When you're counting, when we get to the end here, you have to count carbons, you have to count reduced NADs, you have to count carbon dioxides. So it's really important that you remember that this happens twice for every glucose molecule. Okay, the next couple of slides, I just go through the steps of the Krebs cycle again in word form. Okay, so have a look over those. This, as I showed you for glycolysis, is the full Krebs cycle. This is what's actually going on in the Krebs cycle. Okay, and again, as I did before, I have put this on for the chemists amongst you, those of you who want to know where all the groups are going, or where all the groups are coming from, and exactly what is going on. Okay, this is exactly what is going on. If this confuses you, skip this slide right now. Don't even look at it. Okay, but it's basically explaining what's going on at each step. So again, like I said, if you're not interested in this, skip forward a little bit. If you are, let's just go through it here. So if we look right up here, this is our acetyl-CoA. Okay, this is the acetyl group here, and we've got this coenzyme attached. Okay, so what happens is this 2-carbon compound combines with the 4-carbon compound, which is called oxaloacetate. Okay, to produce the citrate or citric acid. Remember, 8 on the end is like saying a citric acid. So a citrate, citric acid, pyruvate, pyruvic acid. Okay, so don't be wigged out by those. So here we are, we've got our citrate. Okay, and we've got water going in there basically because this is a hydrolysis reaction. Okay, and here we've got our coenzyme A has been released so it can go back to the link reaction. Okay, so we have got our citric acid. Okay, it's converted to isocitrate, and then isocitrate is decarboxylated to produce carbon dioxide, oxidized or reduced, giving us NAD reduced. Remember, we're always going to write NAD reduced because it's a simpler way of writing it and we don't have to worry about all the hydrogens. Okay, that produces a 5-carbon compound which is called alpha ketoglutarate, just in case you see it. Okay, these are the names. Remember, you do not have to learn these. Alpha ketoglutarate is decarboxylated. It is reduced, okay, giving us reduced NAD. Okay, at this point, you'll notice we've still got coenzyme A kind of involved in here, okay, and it produces a molecule called succinyl-CoA. Don't worry about that. Okay, again, the coenzyme helps enzymes do its job, remember, so it's required for this step, but you don't need to know about it. Okay, at this point this is where the ATP is produced. Okay, when the breakdown of the 4 molecule. Now you will notice that this occurs in this diagram after it occurs in the diagram that I showed you for the WJEC syllabus. Okay, there is some discrepancy as to where the ATP is produced. Go with the WJEC. I will be including photographs of the Krebs cycle produced by the WJEC so you can see that, okay? But just be aware, sometimes you will see it occurring later in the Krebs cycle. Do not worry about this GTP and GDP here. Again, you will see this. GTP is a very similar molecule to ATP, and basically ATP is produced sort of through an intermediate. So GTP is produced first and then ATP is produced, okay? But this is the final product. Okay, and then as I said, we've got a couple more steps. This the succinyl-CoA is changed into succinate. This succinate molecule is oxidized to produce, I think it's fumarate, but I can't actually read that underneath there because this sign has popped up. Okay, this fumarate is then produced, this fumarate leads to the production of malate. Malate is oxidized or dehydrogenated to produce reduced NAD. And then we've got this oxaloacetate again. Oxaloacetate is the 4-carbon compound that acetate can join with. Okay? So as it says here, each reaction in this Krebs cycle is catalyzed by a different enzyme, but in a rather nice way. The WJC want you to remember decarboxylase and dehydrogenase. So the enzyme that catalyzes the decarboxylation reactions here and here, and the enzyme that catalyses the dehydrogenation reactions here, 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 and here. That's all you need to remember. Okay. So, to look at it another way, I am going to attempt to talk over this animation again. This is usually where you will find a pause in the video because I've